You good?
Am I connected?
Yep, I can. Yep.
Very happy.
Thank you.
Thank you very much indeed. Just check that box. Okay, so yes, thanks for the organisers for inviting me here today, and hopefully I'm going to try to tell you about some of the work we've been doing at the Crick, which effectively is integrated structural biology, where we've used quite a lot of cryo-electron microscopy, but also integrated NMR studies into this work as well. So with that, whoops, let's get that right. Well, let me just introduce some of the people. Okay, let me just introduce some of the people who've done some of the work. So my lab is at the Crick, as I said, and we are the macromolecular structure laboratory. A lot of the stuff I'm going to talk today is the work of Beppe, Giuseppe Nicastro, Neil Ball, and Dave Goldston. But we also collaborate with other groups within the Crick who are also structural biologists, and I need to mention Oliver Acton and Tom Calcraft, who are in the group of Peter Rosenthal, and they are very much cryo-electron microscopists. We don't just do this on our own, we have real virologists that help us out. So we have the group of Kate Bishop at the Francis Crick Institute, and also Dirk Lindemann, who's our foamy-virus expert who we work with from Dresden. And what I must say, before we can do any of all this work, all of it really, really depends on the help and the support we get from a lot of these structural biology platforms. So we have our Crick one, the NMR structural biology there. We have the NMR Biomedical Centre at the Crick. We also have done some work, solid state stuff with the University of Florence. And more recently, we've started to use the Highfield facility here, and that's all good. So what I would say is we are structural biologists interested in retrovirology. And we're interested in the retroviral capsid, how it assembles, how it interacts with host factors, and how some host factors inhibit infection. So, with no further ado, I'll move on to the next one. Give us a clue. Oh, it only works on the clicker. I have 3 themes. The keyboard obviously doesn't function. I have 3 themes that I want to try and talk about today. First, I'm going to tell you about the high-resolution structures of some retroviral C8 fullerene shell assemblies. Then I want to move on to a bit of dynamics of looking at the interaction of cellular factors with HIV capsid. And then if there's time at the end, we'll see how we're getting on here. I want to tell you about a more recent study we've done looking at a high-resolution structure of a capsid, but within the context of the virion itself. So I don't know if any of you have ever attended any talks on retroviruses, but you're forced to see this obligatory slide, which is the life cycle. Okay. And that starts here. And what we're interested in is the events 1 to 7, which they're called the early events of the life cycle. And it's where the virus binds to the surface of the cell. The core of the virus is released into the cell. It's trafficked to the nucleus. It's pushed through the nuclear pore, and then it becomes integrated into the cell. And that's how you establish a retroviral infection. This yellow thing here is the capsid core. And that's involved in nearly all of these events. So we're interested in what this capsid does and how it's involved in all the events that get the virus from the membrane and get it integrated. And along the way, the virus has to interact with all of these different factors. So we have factors, FES1, kinesin, they're involved in transporting the viral core down microtubules. We have other factors, IP6 and CYP-A, and they're involved in, we think, in keeping the capsid solid together and stable on its journey. We have further factors at the nuclear pore itself, these nuclear porins, NUC358 and NUC153, and they're responsible for transporting the core across the nuclear pore. And then once we're inside the nucleus, we have this factor called CPSF6, which is somehow involved in targeting the viral core and its contents to the chromatin in order for it to integrate. So there are the factors that sort of help viruses to get into cells. But luckily, well, not luckily for everyone, but lots of cells also have defense mechanisms against this. And these are called restriction factors. So we have TRIM5 alpha and TRIM-CYP, and they work by binding to the capsid and somehow disrupting its journey. Further on in the cycle, we have some more factors, FV1 and this MXB, and they also interact with capsid, but they somehow prevent its nuclear passage across the nuclear pore. And there's also some other factors, this APPEC and SAMHD1, which work slightly differently. They don't interact with the core. But on the whole, cellular factors that inhibit the early stages of retroviral infection work through targeting the retroviral core. So why are we interested in cores? And I think I've just sort of summarized the things on this slide here. So basically, the core itself contains the capsid, the RNA and DNA genome, and all the enzymes that are required for a productive infection. So it's got all of the components inside it that the virus needs. In addition, all the critical events, early events, like reverse transcription when the virus has to do, formation of the PIC, they also take place within the core. In addition, an assembled core facilitates the interaction with all of these host factors that are required on its journey from the cell membrane to the chromatin. And on top of that, the core and these critical events are what our cells use to target viral infection or to inhibit viral infection. So I thought I'd just try and show you what some of these cores look like. These are genuine bona fide HIV-1 particles, and the core is this conical dense structure that I hope you can see at the center. We take a bit more of a high resolution image of that. You can start to see that within the cores, there's all this periodicity. And if you take a Fourier filtered image of that, or this might actually be a tube, you can see it consists of this network of interlocking hexagonal structures. So that's a sort of ultrastructure of what they look like. On a more molecular basis, the core is made up of the capsid protein. A capsid monomer contains a CA-NTD and a CA-CTD, so it contains two domains, domain 1, domain 2. And these monomers pack together to form the hexamers that I just showed you on the previous slide. And they do that by the CTD of one monomer packing onto the NTD of the next adjacent monomer in the ring, and so on and so on and so on until you build a disk. The disks then themselves assemble through further CTD-CTD interactions, and they assemble into the cores that we see in the center of the virions. And quite luckily for us, sometimes we can also induce assemblies of these capsid protein in vitro that allows us to study what I would say are very good surrogates for genuine retroviral cores. Just one more thing that I sort of need to add. Depending on the type of virus, the cores all have different shapes. So MLV is a type of gamma retrovirus, and they have spherical cores. MPMV is a beta retrovirus, and they have sort of capsular cores. HIV, as you've just seen on the previous slide, has these conical cores. And this one here, which is only infectious on a Saturday afternoon, has a spherical core as well. And this is important because what I'm trying to get, the core morphology is virus specific, but the underlying hexagonal arrangement is common. And what has to happen is to close these structures, you need to insert into that hexagonal lattice, probably preaching to the converted in this audience, 12 pentameric 5 pentamers into the hexagonal lattice to close the shell. And that is sort of typified here. We've got 12 of these pentamers inserted plus 20 of the hexamers, and that gives us the football shape. And that's the same with all these, there's just more hexamers in them. Done it again. So first tale of horror, let's tell you a little bit about high resolution structure of some retroviral CA shell assemblies. So this all stems because we had the question of, well, we've worked on lentiviruses, so these are all the viruses that infect humans, or exogenous and endogenous retroviruses that are found in humans. So you have lentiviruses that I'm sure lots of people have already heard of, and we have delta retroviruses. These are exogenous viruses that are circulating in the population today. But in addition to this, we also have spuma retroviruses, which are mainly found in primates, in chimps and in gorillas, but they can be zoonotic transmission into humans. And then we have these other two classes, the gamma retroviruses and the beta retroviruses. And they aren't exogenous in humans anymore, they're endogenous viruses. So we wondered, is there a difference between an endogenous retrovirus and an exogenous retrovirus? We chose this HERV-K group for these reasons. So they're the most recent acquisitions into the human genome. They've only been in about 1 million years. They maintain intact open reading frames, and provirus expression of these endogenous viruses associated with inflammatory disease, cancers, and HIV-1 infection. So it's on HIV-1 infection, their expression is induced. And it was possible by some other groups that they were able to reconstitute a consensus HML2 sequence and make viruses that were processed an infection. And here's just some images here of some of the viruses budding from cells. So the question is, how does the CA structure from an endogenous retrovirus compare or relate to that of an present-day exogenous virus? Okay, so very quickly, the first thing we did is we determined crystals and NMR structures from the domain. So this is an X-ray structure of the N-terminal domain of the HML2 capsid protein. So basically, it's this helical, 6 helical bundle with a beta-1, beta-2 hairpin. We also determined this time by solution NMR the structure of the C-terminal domain, and that has this 4 helix bundle, and it's linked, they're associated with each other through this dimeric interface, which involves this alpha-8, alpha-8. Now that's reasonably quite, what's the word? The dimer itself is incredibly weak. It's got a KD of about 275 micromolar, so we were pretty lucky to be able to determine the structure of something so weak. But what we were able to do is we were able to carry out this filtered experiment where we could just detect NOEs between monomer A and monomer B. And we did that by having C13, N15 filtered nosy spectra that just left us with the 12, 13 NOE couplings across there. And armed with that, we're sort of very confident that we know that this dimer interface is real and we have a good C-terminal NMR structure, and that's important for later on. Okay, so in the course of doing this, we also wanted to look at intact capsid. And every time we sort of made the protein, we got this persistently annoying high molecular weight material that contaminated our preparation. So this is our nice monomer dimer capsid protein, and this stuff just, we could never get rid of it. So eventually we decided, well, let's have a look what it actually is, and we carried out some light scattering experiments, and it told us that we had stuff in there in the range of 1.6 to 3 million Dalton molecular weights, and a bit more high resolution experiments using sedimentation velocity, we could find 32S, 42S, and 50S particles that were all within this heterogeneous peak. So given we'd seen that, I guess it was impossible for us not to at least look by cryo-electron microscopy what was actually in there. And this is a field of particles that came from that gel filtration peak. And you can see it's absolutely stuffed. And you'll have to believe me, there are 4 types of particle in this field. And this is the FSC, this is the limit of resolution for structures that we've determined for these particles. So the first one we've got a T is 1 icosahedral particle that contains just 12 pentamers, and we have that at 2.7 angstrom resolution. We've got a D5 capsular particle, and that's got 12 pentamers and 5 hexamers. We've got that at about 3.5. We've got a further capsular particle, a D6, which contains 12 pentamers and 8 hexamers, and we've got that at about 3.8. And the best one of them all is we have a T3 particle. That is the same as the football that I showed you before. That contains 12 pentamers and 20 hexamers. And these structures, by analyzing them, should allow us to visualize a high resolution, all of the interactions that make up capsid shells. So if I take them one at a time, this is the T is 1 particle. So we've got this at about 2.7 angstrom. This is, I've just ringed some of the ones here. You'll have to believe me that that's actually one of these, but I assure you the computer software can pick the right particles. So that's made of these 12 pentamers here. Here, the red is the NTD and the N-terminal domain, and that forms this pentameric shape. And then the pentamers are then linked together through C-terminal interactions between this underlying cyan layer, which is the C-terminal domain. So that's the T is 1. The D5 still has 12 pentamers, but now they're arranged in caps at both ends, and then that's interspersed by a belt of 5 hexamers this time. So again, we have the same underlying CTD lattice, but now we've got pentamers displayed at the top and the bottom, and hexamers displayed around the middle. Our D6 is again similar, but now we have 6 pentamers at the top, and now the pole contains a pentamer, 6 pentamers at the bottom with another hexamer at the pole, and then this time we have a belt of 6 hexamers around the center of the particle. And then finally, the T is 3. And again, they all have this same underlying CTD lattice, but this one has our 12 pentamers, but now they're interspersed all the time with hexamers. So there's no actual pentamer-pentamer interactions. There's only pentamer-hexamer interactions and hexamer-hexamer interactions. So just to assure you of what I'm saying, so this is the sort of monomer structure, and this is just a piece of the electron density that I've taken out of the cryo-EM map. And what you can find is we have the resolution to certainly say we can see the side chains and we can be confident in the sort of interpretation that we're saying. And the section from about this 143 down to this 158 was actually missing from both of our structures. So we built this one in de novo. We didn't have that in our X-ray structure or in our NMR structure. And then over on this side, that's just looking at some density for the CTD interface, and you can see again we've got good side chain density here, so we're confident in our interpretation of what we're going to say about C-terminal dimers. Okay, so we've got a wealth of information. There's lots of questions we could ask. There's lots of questions we may be able to answer. So I guess one of the first ones is how do you get from a pentamer to a hexamer? So it's actually quite simple. All you need to do, we've got this ring of alpha 1s around the center. It's just a very small rotation of the alpha 1 and a little tilt, and then we're able to then open up the pentamer and then insert another monomer from the hexamer to generate our hexameric structure. And I guess the easiest way to see that, and here we go. How will it work? Give us a clue. You do it. That's it. So basically that's the hexamer. You take one out, you tilt it and you fold it around, and we just run that 3 times. And again, you can see all you need to do is do that. So it's all accommodated just from what goes around in the center of the disk. So we move on. Next one. Okay. So that's the sort of NTD interactions. But what other differences are there between pentamer and hexamer? So we move on to what we would call our NTD CTD interface. That's shown here. So this is how one monomer packs onto the next monomer going around in the ring. And what you find here is you have this sort of network of hydrogen bond interactions. So you have this R143 K182 interaction between an NTD and a CTD. And then you have this set, or sort of this range of hydrogen bonding interactions between the CTD of monomer 1 and the NTD of monomer 2. So that packs on there, and it's got this characteristic 181, 79, 171, 90. So that's the pentamer arrangement. If we then move on to the hexamer, we find that, again, we have a similar set of hydrogen bonding interactions, but they're not quite the same. So now R143 is moved from its position here onto the next residue. The 79, 171 has shifted a bit more, and then we've broken the 9219. So we have this toggle. So basically, to move from the pentamer to the hexamer, all you have to do is just toggle around these, move slightly and toggle around these 3 or 4 hydrogen bonding interactions. So that's the way the protein can adapt to these 2 different oligomers. Okay, so then moving on to our T is 3 hexamer, you'd think it would all be the same, but it turns out it's not quite the same, because each pentamer sees hexamers and they're all identical. But hexamers see both pentamers and other hexamers. So what that creates is it breaks the C6 symmetry. So they're not really hexamers anymore, they're actually trimers, where 3 of the subunits interact with hexamers and 3 of the subunits interact with pentamers. So we've got non-equivalent interactions. So what does that do to our toggle? Well, here we go. So what we find here is that in a T is 3 hexamer, when the subunit is interacting with neighboring pentamers, it adopts the toggle interaction as if it were a pentamer interaction. When it interacts with neighboring hexamers, the toggle, it's not quite exactly the hexamer one, but it's sort of intermediate between both. So you've got, in this way, you can adapt to not having perfect C6 symmetry. You can have breaks in that symmetry by, again, just changing that toggle. Oops. Okay, so that's the sort of NTD, CTD sort of stuff. What about the C-terminal dimer? So now we can move on to our solution NMR structure we have here. And this is just showing the sort of cartoon representation of the dimer. And basically, it's largely made up of hydrophobic interactions between the residues on these crossing alpha-8 helices. And what you have to notice is they run more or less parallel to each other in the solution structure. When we looked at CT dimers, however, in particles, we had a bit of a surprise, shall we say. So when we looked at dimers that connected pentamers together, we found that the helices no longer run parallel to each other. The hydrophobic interface is sort of maintained, but now the cross angles move up to about 60 degrees. And in a hexamer-hexamer interface, the crossing angle is even wider. It's now more like about 90 degrees. So you get a rearrangement of some of the salt bridges on the end and a loss of these hydrophobic interactions when this dimer is incorporated into shells. So what we can do is we can take the few hundred that we've got in all our shells, and we can take the 20-odd NMR structures. So that's the NMR dimer, that's a pentamer-penta dimer, a hexamer-pentamer dimer, and a hexamer-hexamer dimer. And we can classify them into different types by sort of plotting their displacement against their angle. And we call this a ball plot, not because they're balls, but because that was the name of the guy in my lab who did them first, Neil Ball. So what we have here, all the NMR structures all group in what we'll call class 1. So they're all about crossing angles of about 20 degrees. All the pentamer-pentamer and pentamer-hexamer interactions, they group with this central class 2 of about 60 degrees. And then the class 3 hexamer-hexamer dimers, they all group with this other 90-degree crossing angle. So the solution state dimer, I'd say, is probably the most stable, but it appears it's not found at all within the context of the viral shells. And we have this quantization of either 60-degree or 90-degree angles, depending whether you're at a steep part of the shell or a shallow part of the shell. So the CTD interface can adapt to accommodate shell curvature. So I think just to summarize everything I've gone on about there, HML2 capsid can form fullerene shells in vitro, and we've been able to determine 4 classes to pretty high resolution by single particle cryo-EM. You get small NTD tilts and rotations, as well as toggling of NTD interactions. And that's how you can rearrange a pentamer into a hexamer. The T is 3 hexamers break the 6-fold, and through NTD CTD toggling can interact with either neighboring pentamers or neighboring hexamers by adopting the arrangement that's pentamer-like or hexamer-like. And then at the C-terminal domain, pentamer, hexamer, and hexamer interactions are distinguishable by the quantization of their crossing angle, and they're all very different from the non-strained state we see in solution. So overall, shells, we have hexamer, pentamer interfaces, curvatures accommodated by small rotations at the NTD, toggling shifts, and large domain movements. And that, I think, gives us a sort of rule book for how you can build a capsid that can accommodate steep places where you've got 5-folds and shallow places where you've got runs of hexamers. Okay, so that was sort of number 1. So I thought perhaps we could move on to number 2, and I guess you're desperate to see some NMR. So what we've got here is where we looked at the HIV capsid shell and its interaction and the dynamics of the shell upon interaction with cellular factors. And we chose here, to go back to our tree, to use lentiviruses. So this time we're using HIV capsids, and it's not just the obvious health interest in lentiviruses, but they have this property that the HIV capsid in vitro, we can very readily assemble them into these sort of tube shapes here. We can label them, N15, deuterate them, do anything we want with them. But even better, we can pack them into a MAS rotor and we can generate spectra like this. So this is a short mixing time DAR, and this is a sort of NCA correlation spectrum. And taking these spectra plus a load of 2Ds, 3Ds, some of the solution assignments transfer and the stuff in the literature, we're able to get assignments for getting on for 90% of the backbone, and I think some of the betas, carbons, and nitrogens for the whole thing. That gives us the capability then to say, well, now we can add ligands to these things and we can see what effects these ligands have on our surrogate for a retroviral shell for a retroviral shell. Before we did that, we chose these two ligands here. So I mentioned right back on the first few slides, I mentioned one called IP6 and one called CPSF6. So IP6 looks like it's involved in stabilizing the capsid shell so that when it goes into cells, it just doesn't fall apart. So there's literature to show that. Then the CPSF6 peptide, as I said, is involved in helping transport capsids once they get into the nucleus towards the chromatin for integration. So we solved a ternary crystal structure first, and that shows IP6 bound down the middle and a nice belt of CPSF6 molecules bound around the outside. And the reason we did this is so will help us interpret some of the NMR data that I'm going to show you in a minute. So just to take this IP6 structure, what you can see is there are actually two molecules of IP6 bind right down the central channel of the disk, and they basically interact with sort of rings of basic residues. So here I've just highlighted these arginine 18s, but there are also lysine residues in here as well, which bind to the IP6. CPSF6, slightly different. That sits between an NTD and CTD in a pocket, and it forms this sort of horseshoe structure here, and that interacts with many of the hydrophobic side chains that are exposed on the surface of these two helices here. So the N57, L56, I73, I69. And if you make mutations in any of these residues, you reduce viral fitness. And also, as an aside, this is the place where a lot of capsid drugs have been targeted to do. So that's another story I can tell you if anybody's interested. Okay. So then we collected a load of solid state data where we basically looked at what the effect of adding IP6, ATP gamma S, which is like a little bit like IP6, and then CPSF6 to these tube assemblies. And then we looked what happened to the chemical shift, any chemical shift perturbation and or changes in the peak intensity sort of ratio between the free and the bound. And to cut a long story short, the IP6 we found has some very localized chemical shift perturbation changes, but what it did do is it appeared to cause quite large scale intensity changes across quite a broad part of the whole protein. And in particular, it's in the interfaces between NTD, NTD and CTD, CTD at the edges of each disc, which are where the sort of discs are linked together to form the shell. By contrast, CPSF6 peptide didn't seem to have very many global changes, but it had quite a lot of local changes. So all of the residues very close to that binding site that I just showed you in the crystal structure were all had big changes in chemical shift, as you might expect. And we interpret this as that the IP6 binds in the center of the disk, but then it's by transmission of its binding, by allosteric sort of transmission, you could say, its effects are felt as a stiffening and or a rigidization of the whole shell structure. And that's probably what we think where the stabilization effect of the molecule comes from. On the other hand, CPSF6 just sits into a preformed pocket and just binds there and doesn't have any big effects across the whole shell itself. So we're a bit intrigued by the IP6 effect, and to sort of strengthen it a little bit, we decided to carry out a selective labeling experiment. So what we have here is an NCA where we've just labeled all the lysines in the protein. So we've got lysines 20, 30, 25 and a few here. So we have a very much simplified spectrum, and of course we can assign it quite nicely. Then when we add IP6, what we find is we get all these, well, we get these spectral changes. So some things don't change at all, but quite interestingly, the residues close to the IP6, they show chemical shift changes and they move, and then this one strengthens. And then, but more interestingly, these residues down here, K199 and K203, which are 50 angstroms from the binding site, they show chemical shift changes as well. And in particular, this 203, which is right in the threefold axis of the shell, that goes from one resonance, which is coalesced in the unbound, to two separate resonances. So it's like two conformations become frozen out. Again, more evidence of the sort of stabilization, rigidification of the structure. So to sort of summarize what we've found about that. So we think IP6 and CPSFs can co-occupy HIV capsids. So I guess when we were doing this work, one of the sort of theories was the idea that you get a handoff within cells where IP6 would stabilize a thing when it went in, and then that would be handed off onto another factor and then another factor. But in our hand, both of the molecules bind to discs, and there isn't any obvious sort of allosteric changes as a result of one binding over the other one. So the data is largely additive. IP6 induces small chemical effects around the surrounding residues, but larger allosteric transmission and the dynamics of distal residues at the interhexamer, NTD, and interhexamer, CTD interfaces. And that's consistent with it acting as this capsid stabilizing factor. Binding of CPS6 has smaller global effects, but induces chemical shift perturbations in the residues surrounding it. And that's consistent with tight specific binding to this preformed site. And then we have further evidence using the selective labeling experiment that IP6 can induce change in the dynamics of that lysine at the CTD trimer, where it freezes out two conformations from something that's coalesced in the non-liganded structure. So that's good. So what I thought the last thing we can tell you about, so I've done in vitro assemblies, we've done some dynamics of capsids, but now we want to move on to something where we've tried to look at an in situ cryo-EM structure of the actual capsid, but actually within the context of the virus. And we've done that, and we've done that this time in using this type of viruses, the spuma retroviruses. Now, these are true retroviruses. They largely infect primates, as I said, but they are found in humans, and they're not pathogenic. So there's great interest in using these things as retroviral vectors or in gene therapy and or oncolytic viruses. And what's unusual about them is they have a very different sort of gag protein to the orthoretroviruses, and they're illustrated on this slide here. So in the normal course of events, in an orthoretrovirus, you have the gag protein, and it's cleaved by in the process of maturation into three proteins, P17, P24, four proteins, P7 and P6. And these three proteins here go on to form the ultrastructure inside the virion. So this is called the matrix, which is near the membrane. This is the capsid or the core I've been talking about. And this one binds the viral genome. In foamiviruses, you have equivalent regions. You have an NC, which is this GR, a capsid region, and an M matrix region. But in foamiviruses, the gag isn't cleaved. So all of the structural domains remain within gag in the assembled virion. But there's one small cleavage here at the C terminus, which gives you this P71 and P68. Both are required to be infection. But the main thing here, all of the structural domains are maintained within gag. They're not processed out. There is no maturation process. So we determined this one's an X-ray structure of the gag internal domain, and this should be the equivalent of the matrix of the orthoretroviruses, but it turns out it's not related at all. We're not sure why, but it's got absolutely no relationship whatsoever to matrix of ortho. It's got a coil and these head domains, and basically they're required for the virus to be picked up by its envelope to egress cells. And these little peptides here are a complex we solved of the N protein bound to the head of the matrix. And then just to say, it's also the target for the TRIM5 restriction factor that I talked about right at the very, very beginning, which is usually capsid, but in this case, it turns out to be the matrix. Additionally, we have an NMR structure, and this one is in solution of the capsid domain. And again, we have this N-terminal domain and C-terminal domain. This time, we can only find the monomer in solution. And the main thing to say is that they're packed a lot more tightly, or a lot more closely together in the foamiviruses than they were in the orthoretroviruses. We also looked by EM at the cores, and what you can see, they're quite regular indeed, right? So that is just a drawing over the top of that. So we have a very regular core inside the foamivirus, unlike the sort of big fullerene shells we saw in the orthos. And if you make mutations in any of these residues, these are NOEs from NTD to CTD, you find that you disrupt the cores, and yes, you might have thought these viruses are uninfectious. So given we could image these viral particles quite easily, I mean, that tempted us to say, well, can we get any much more higher resolution structural information? So we took 9,000 of these cores, there and there. We just basically masked out the center of the virion, the actual icosahedral thing, and then we were able to solve the structure of these cores in the context of the virion to about 3.5 angstroms resolution. And that's what it looks like. So basically, it's a regular Ts13 icosahedron. It's about 600 angstroms in diameter. And we were able to build 780 copies of our PFV CA monomer into this, and it constitutes 12 pentamers, 720 hexamers, and that, if you want to know, is a molecular weight of 20 million Daltons. So why was that made up? So this is the asymmetric unit. So the asymmetric unit, as I said, this is a Ts13 particle. So it consists of 13 CA monomers. There's one here that comes from a pentamer because these are all identical. And then we've got 12 that are distributed between two hexamers, hexamer 1, and hexamer 2. Now, on the sides of it, they're hexameric, but as I pointed out with HML2, things aren't always quite that simple. So this hexamer's only got twofold symmetry, so it's broken down here. And this hexamer has no symmetry, no cyclic symmetry whatsoever. All 6 copies have slightly different environments. And here what I've done is we took all 13 chains and then we align them against each other to see if we could find classes. And we find a red class, a blue class, and a green class. And it turns out the red class isn't even unique to the pentamer. It turns out in the pentamer, it turns up once in hexamer 1, once in hexamer 2. And then the blue and the green classes are 2 times in hexamer 1 and 2 and 3 times hexamer 2. So what's the origin of that? So then we did this sort of cluster analysis here. We took each N-term, each capsid domain and looked to see how they related to each other. And we came up with these 3 classes, which we call loop term, loose loop, and tight loop. And what that pertains to is, and the NMR structure this time, indeed, isn't out on its own. It belongs to this tight loop class. So this one is actually found in the shell. So the difference between the things is just the conformation of the loop that links the N-terminal domain and the C-terminal domain together. What the difference is that it slightly alters the pose of the NTD with respect to the CTD. And additionally, it changes the configuration of the residues that are presented on the outside of this loop. And they're involved in the packing against neighboring monomers within the Ts13 asymmetric unit. So what that allows it to do, you can build this 13-mer jigsaw piece that can accommodate all on its own, where the 5-fold is very steep, and then where the 6s onto the 6 is very shallow, all just by changing the sort of amount of where these 3 different types of the monomer are placed within the Ts13 asymmetric unit. So, putting this into context, what we then thought we could do is, because we've got these high resolution structures, can we put them all back within the context of the whole particle? So what I've got here, this is the image I showed you before, and these are sort of particles where we've reconstituted the proteins back onto these particles. Now, I haven't talked about this. These cyan and pink things, these are the envelope proteins, which isn't my thing, but we're able to place those, and they form this sort of surface lattice or matrix on the surface of the virion. And then within the middle, we've got the capsid that I've been describing to you. And then if we take these particles and do some sort of radial density averaging, we can look at all the layers of organization within the virion. So right at the middle, we've got the nuclear capsid in the genome that forms this reasonably diffuse layer. Then we've got a very strong layer here that comes from the GAG capsid itself. And then we've got 2 other peaks. We've got one near the membrane at 500 angstrom radius and one inside at about 400 angstrom radius. The 500 corresponds to the envelope protein, this matrix here. And they've got this one in the middle that it obviously isn't ordered enough that we could see it to basically get a map good enough to build the structure. But it still forms a sort of semi-amorphous or semi-structured layer at about 400 angstroms. And we believe that must correspond to our GAG N-terminal domain that I showed you before. So to summarize what we see on that. So basically PFV forms this regular Ts13 capsid. It's got fullerene geometry, unlike what you see in the orthoretroviruses. We've got 13 monomers in the asymmetric unit, 1 from a pentamer, 12 others from 2 types of hexamer. Both of them don't have cyclic symmetry anymore. We've got 3 classes of intramolecular CTD, NTD pose. We've got loop turn, tight loop, and loose loop, and they direct the packing of different side chain configurations of the loop to basically bind to neighboring protomers. And then all of this structural plasticity at NTD, intracapsular NTD, NTD, CTD, and intercapsular dimeric and trimeric interfaces allows for the formation of this TS13AU that allows us to build this massive icosahedron. And then finally, we reconstituted the whole virion and found its layers of organization. We have a highly ordered capsid, N proteins that form a surface lattice. And then we got this amorphous layer between M and cap. And we think that probably functions in target cells, possibly to encase the capsid, because it's also what's recognized by the trim restriction factor. And with that, I just want to, again, thank everyone who's been involved in all the work. These are all the Crick people, all the people from the facilities. And of course, I've got to say, acknowledge our funders, without which we couldn't do any work. And I should acknowledge everyone in the audience, and I'm happy to stop and take any questions.